Phil came to KCU after graduating from University of Nevada in Reno in 2016 with his BS in Biochemistry and Molecular Biology. In Reno, he worked as a hospital phlebotomist and clinical lab assistant while also volunteering his time at a clean needle syringe clinic. There, he provided addicts with a source of sterile needles, counseled clients, did anonymous STD testing, and most importantly, provided education on poverty and drug addiction, including where and how to get help. Since joining KCU, he has been third author on the paper Cadaveric Analysis on the Spatial Properties of the Sural Nerve around the Lateral Malleolus, which will be presented at KCU's Research Symposium in April. He's also been a logistics coordinator for the EM Club, and he's been a COVID vaccine volunteer. So again, thank you for joining us, and I'll turn it over to you, Phil.
All right, thank you so much. I'm going to turn the screen share on. Okay. All right, so okay, so I want to thank everyone who's joined. My name is Phil Vayner, and my presentation is on a report on 2 successful attempts at treating beta thalassemia and sickle cell disease using CRISPR-Cas9. Okay, so to start, I want to give everyone a quick review of sickle cell disease, beta thalassemia, and a general overview of how CRISPR is used in gene editing before we actually dig into the paper itself. So we'll start with sickle cell disease. Let me turn a laser pointer on first. So the epidemiology of sickle cell disease is that it primarily affects individuals of African and East Mediterranean descent. In the United States, 8% of our African American population are carriers of the sickle gene, and it's considered the most common intrinsic hemolytic anemia worldwide. And a bit on the genetics and pathophysiology of sickle cell disease. So the patho, it's heavily dependent on the actual genetic makeup of the individuals. So in a heterozygote, for instance, they'll be a carrier of only one defected allele, and so they'll actually usually be asymptomatic, and the heterozygote genotype is actually considered, or is believed to be, to confer protection against malaria. Now, the problem really shows up with homozygotes that have both sickle alleles, and they'll end up with the very brutal symptoms associated with sickle cell disease. What happens is there's a point mutation in the beta globin gene on chromosome 11, and it leads to a switch between the glutamic acid side chain switching to a valine side chain, and that produces the sickle hemoglobin phenotype. And then when you have that sickle hemoglobin phenotype, what happens is whenever the person is under any hypoxic stress, be it high altitude or exercise, those as hemoglobin sickle. And in a future slide, we'll have a picture of a microscopic slide that shows what the sickling is, what the sickling looks like, but I'm sure a lot of us have already seen those microscopic slides, so it'll just be a little review of what they look like. And when they sickle, the patients end up experiencing like a very debilitating vaso-occlusive event, and it severely affects the quality of life for the patients. And those vaso-occlusive events lead to potential tissue infarction due to ischemia and decreased oxygen. So here is that picture I was talking about. I have more later as well. Right here, you can see a sickled hemoglobin, another sickled hemoglobin. And yeah, it's these shaped hemoglobin cells, these shaped red blood cells that lead to the vaso-occlusion. Now here's this table that I put into the presentation to show the composition of hemoglobin in normal patients without any sickle cell alleles. A patient with sickle cell trait, meaning they're heterozygotes, they only have one, and then the patients that are homozygotes and they have both. You can see here, the normal has no sickle hemoglobin, no HbS. Then when you introduce one allele, you get 40%. And then when you introduce both, it's predominantly hemoglobin S. Now, what's interesting And what I'm going to touch on a lot more later in this presentation is what we see here with the HbF, which is fetal hemoglobin, and how it's remarkably increased in patients with sickle cell disease. And the understanding is the greater the expression of fetal hemoglobin, the more attenuated, the lesser the symptoms of the sickle cell disease will be for the patient. And for a while now, researchers have known that if we can find ways to induce an increased level of fetal hemoglobin in their patients, they'll be better. Their prognosis, their treatment will be better in sickle cell disease. And one way they've been using, one thing they've been doing is hydroxyurea. That's been considered a first-line treatment for elevating fetal hemoglobin. But again, we'll talk about this more later in the presentation. Oh, and currently, well, besides for this use of CRISPR that hasn't been generalized, the only real curative therapy for sickle cell disease is an allergenic bone marrow transplant, where we're taking cells from someone else with a similar HLA antigen matching and then infusing, grafting, transplanting those bone marrow cells into a patient. But the problem is there's only a 20% chance of finding an eligible patient, or there's only a 20% chance that a patient will find an eligible donor. So this doesn't work out for the majority of people with sickle cell disease. Here's another picture. Here's a pretty classic sickled hemoglobin. Okay, and the next disease that was treated in this paper is transfusion-dependent beta-thalassemia. The epidemiology is that it mostly affects people of Mediterranean descent, primarily people of Greek and Turkish descent. The allele is pretty rare in the US actually, but 7% of carriers, 7% of the world's population are carriers. So in other parts of the globe, it's a lot more prevalent. And what happens here is it's just a point mutation somewhere in the promoter sequence of splicing sites in the beta globin locus, also on chromosome 11, just like sickle cell disease. And it can produce two different variants. The beta positive variant is when there's one defective allele. Symptoms are unremarkable, kind of in the same way it is with sickle cell disease, where you really need to have two defective alleles, the beta not phenotype, where you begin to see really, really poor prognoses and difficult symptoms for the patients. And what happens when there's these defective alleles, there's an imbalance between the alpha-like and beta-like globin chains in hemoglobin. So you end up with ineffective erythropoiesis and increased hemolysis. And again, the curative treatment is the same. It's an allogenic stem cell transplantation, but that's not always available for everybody. So better treatments would be a lot more beneficial, or treatments that are more generalizable that more people can have access to is an ultimate goal. Okay, so now let's talk about CRISPR. So the reason I included some of these images in this intro slide is that because there's a lot of noise surrounding CRISPR, and a lot of times this noise really shadows the potential that CRISPR has today in treating a lot of genetic diseases. You'll hear about designer babies, or what happened in China when they decided to perform CRISPR edits on actual embryos. So I felt really inspired to do this presentation to show that there are clinical uses that are ethical, that are available to us now, that should be the focus on talks when it comes to CRISPR. Okay, so here's a brief overview. The next slide I have is actually a schematic. It'll be easier for me to just show step by step with the pointer how the CRISPR-Cas9 system works. But here I'll just mention that it's actually derived from prokaryotes, and in prokaryotes, it's an immune defense system, whereas we're going to be using their immune defense system as a gene editing tool. And with that gene editing tool, we can perform gene knock-ins, knockouts, gene silencing, gene activation. There's a lot of capabilities with CRISPR. Okay, so here's the schematic of CRISPR. I'll start at the top with Here would be our DNA sequence of interest. This is where we want to perform an insertion, a deletion, a silencing. Here we have the guide RNA, which is synthetically derived, meaning we'll make it in a lab through some methods, and it'll be attached to this Cas9 protein. The Cas9 protein isn't very well visualized in the schematic, but imagine that this were a protein, there's like a boundary around it, and it's carrying this guide RNA, and they move to the actual DNA sequence of interest. The Cas9 protein is an endonuclease, and it's going to perform a double-stranded break once the guide RNA is brought to the gene of interest. And then from there, there are a lot of options in how researchers can perform edits. The most simple would be the non-homologous end joining method, which is also considered using wild type Cas9, CRISPR-Cas9, where it's not actually going to be very accurate. The Cas9 protein is just going to start making disruptions in that sequence overall. So they're not going to be, if they're going to be employing this technique, they aren't really worried about specificity of disruption or editing within the DNA sequence of interest. And that's actually what they used here, which is pretty remarkable considering we're doing this to actual human cells. And I'll touch on why that's remarkable and why it works later in the presentation. And then here you can see a correction of mutation, and that's from homology-directed repair. Here, insertion of a normal gene copy through homology-directed repair. And this is a different Cas9 system, CRISPR-Cas9 system, that'll just catalytically impair the DNA sequence just by introducing a thymine in place of a cytosine. Okay. All right. So now that we've covered the essential components behind this paper, we're ready to dig in. This paper, it goes over the successful curative CRISPR-Cas9 therapy 2 patients received. One of the patients had sickle cell disease and the other had transfusion-dependent beta-thalassemia. The paper goes into detail on the development of this cellular drug, which I'll touch on, called CTX-001. The preclinical testing of CTX-001 to ensure that it's going to be safe before they actually infuse CTX-001 into 2 patients, and then the 21 months of regular follow-up with the patients. Okay, so I'm going to actually touch on fetal hemoglobin one more time, a little bit more in depth, and this is the main concept behind CTX-001 and how they use CRISPR to handle both transfusion-dependent beta-thalassemia and sickle cell disease. So there is a gene called BCL11A, and its expression increases as we near the moment of birth and then greatly increases within the first 3 months. As expression of BCL11A increases, expression of gamma hemoglobin decreases. Gamma hemoglobin being the primary component alongside alpha hemoglobin in fetal hemoglobin. As this decreases, eventually it'll reach a threshold. Here we see it around, I'd say that's 18%, where the onset of symptoms begins. So with CRISPR, they wanted to find a way to inactivate BCL11A to prevent this decrease in expression of gamma hemoglobin. And this is that slide again, just showing that in sickle cell disease, you see that they have that range between 5 and 25%. So those that have the 25% upper threshold really don't have too remarkable of symptoms even with sickle cell disease. But when you're closer to 5%, it's pretty drastic. Okay, so now I'm going to talk about what is CTX-001. CTX-001 is an autologous cellular drug, autologous meaning that the cells are derived from the same patient receiving the cells. They're derived from CD34 hematopoietic stem cells and progenitor cells that are collected by apheresis. So yeah, they would take the patient's own cells, they would extract the CD34 cells from those cells after performing the apheresis, and then they'll treat the cells with CRISPR-Cas9 to enhance induce edits into this region right here, the erythroid enhancer region of BCL11A. And on the next page, I actually have another schematic on the manufacturing process of CTX001 and then the infusion process into patients. And then, yeah, once they perform the edits, they will implant the cells in a manner similar to a bone marrow transplant, where they'll myeloablate the patient, knock out their bone marrow immune system, and then infuse them with their own cells in hopes of engraftment, and then ultimately that knocked out expression and a whole new set of blood cells, essentially. Okay, so here's that schematic, kind of how the whole process would start. It would start right here. You have your patient with some chronic blood disorder, in this case, sickle cell disease or beta-thalassemia. The cells are collected via mobilization and apheresis. So here we have our cells. They're going to get shipped to the manufacturing location where they're going to take the cells and they're going to isolate just the CD34 positive cells. Then they're going to electroporate CRISPR-Cas9 into the cells to perform the edits. At this point, these are the autologous cells that are edited, and they're now considered the drug itself, CTX001. They're going to send the CTX001 back to wherever the patient is stationed. Now the patient is going to go under the myeloablative conditioning to knock out their bone marrow, knock out their immune system to prepare for the infusion. Then they're going to be infused. We're assuming that the cells will engraft correctly and the immune system will redevelop, and then they'll just follow the patients for 2 years, up to 15 years, and then, yeah, so they discharge in 2 years of follow-up. Okay, so before reaching the point where they could actually use CTX in the patients, they needed to perform several experiments to ensure that it was safe and effective. The first thing they did was they pulled CD34 positive cells from 10 healthy donor patients. Then they took those cells and they pretty much plated them, treated them with various growth factors, keeping them functional in vitro. And in vitro, they measured, after performing the CTX edits, the expression of fetal hemoglobin. And what they saw was the edited cells had a 29% increase in fetal hemoglobin production, or no, 29% of fetal hemoglobin relative to total hemoglobin. And then the controls, the unedited cells, only had 10% fetal hemoglobin relative to the total amount of hemoglobin, total composition of hemoglobin. Okay, so they verified that the edits worked and that the edits would increase the expression of fetal hemoglobin. Next thing they had to do was ensure that those cells would actually engraft into, sorry about that. They had to make sure that the cells were actually engraftable in an organism, so they used mice. And what they found was when they took 3 samples of edited CD34 human cells and they engrafted them into mice, the mice went under myeloablative conditioning, prepared for an infusion of bone marrow. And after a 16-week period, though the mice showed an allelic editing frequency of 91.4%. Prior to that, the cells themselves showed a 95.1% allelic editing frequency. So they performed something called a one-sided Mann-Whitney test, which checks for a loss of editing after engraftment. And according to the results of the Mann-Whitney test, this 4%, a little under 4% decrease in allelic editing frequency was not considered a significant reduction in editing over the time span of engraftment. Thus, the engraftment was successful in mice after a 16-week period. And lastly, before infusing into patients, they wanted to ensure that the edits were actually accurate, because it's one thing to have an edit take place where edits to take place where you want it and where you don't want it. So to do that, they took two approaches simultaneously. First, they performed sequence similarity analysis of the entire genome to find as many locations in the genome that seemed close enough to the BCL11A genome to be considered a potential target for an off-site edit. The way they categorize that would be any site with 3 or less mismatches, or any site with 2 or less mismatches and 1 DNA RNA bulge. And using those characteristics, they found 171 candidate off-target sites. Then the other way they approached searching for candidate off-target sites was a laboratory-based approach using a technique called guide sequencing, where they just looked for potential sites for DNA double-stranded breaks that had a high probability of allowing the Cas9 protein to do its edits. And they found 52 candidate off-target sites in that manner. And what they ended up finding with the 171 plus 52 was 223 total off-target sites that they believed had a good probability of receiving an off-target edit, but what ended up happening was none of them had an off-target edit, which is amazing. And then the one spot that they wanted that edit to take place at, the BCL11A erythroid promoter sequence, had the edit. So that shows, perfect accuracy in terms of this experiment right here. Okay, so now they consider themselves ready to actually infuse patients with their CTX001. Our patient 1 was the patient with transfusion-dependent B-Thalassemia. The patient had the beta-not, beta-positive genotype. And I know I mentioned earlier that generally this genotype has lesser symptoms, but there's actually a spectrum within the heterozygote genotype, and this patient had pretty remarkable symptoms from the transfusion-dependent beta-thalassemia. She was 19 when she consented to the trial. She's a female. And prior to the study, she received 34 units of packed red blood cells a year, and she would need 16.5 transfusions per year. So like I said, definitely had remarkable symptoms. Following infusion, she had 32 adverse events, 2 severe adverse events, and this was all within the 21.5 months of follow-up. The adverse events were characterized by grades. Grade 1 were the lowest severity adverse events, 2 was increased, 3 was increased, 4 would have been considered even worse than that. Everything resolved on its own, or not resolved on its own, but resolved with treatment. The 2 serious adverse events were pneumonia in the presence of neutropenia and lymphopenia, and veno-occlusive liver disease with sinusoidal obstruction syndrome. The pneumonia, it began on day 13 post-infusion, and it resolved day 28. And the VOD-SOS began on day 13 and resolved day 39. The patient 1 actually had a history of hepatitis C, so it was believed that that was a major reason why the patient experienced this severe adverse event, but again, it resolved on its own. Did not resolve on its own, it resolved with treatment. Okay, so here I'm just, this table shows the engraftment rate in both patient 1 and patient 2. So they infused, so the CTX001 cells infused into patient 1 had a allelic editing frequency of 68.9%. Then after engraftment, the patient's own peripheral blood cells showed an allelic editing frequency of these numbers as follows, where it started at 48.1% all the way to 62.9% at month 18. When they took bone marrow aspirates, the allelic frequency editing was 78.1% and 76.1%. So shows that it's really taken foot in the bone marrow after infusion to really show that the CTX001 engrafted correctly. within the patient. Patient 2 required 2 mobilization cycles. The paper didn't actually go into why the patient needed 2 mobilization cycles, which I thought was very peculiar. Mobilization cycles as in apherese the cells, take the cells from the patient, then perform the edits, then reinfuse cells into the patient, so reinfuse the CTX001, and then repeat that all over again, which is what was considered 2 mobilization cycles. But they did mention that patient 2 needed to be hospitalized during this process, whereas they never mentioned any hospitalization required for patient 1. So that could have had something to do with it, but again, they don't go into why. But more importantly, both patients did end up engrafted with the CTX001 cells. Okay, so here are the results for patient 1. So patient 1 originally needed 16.5 infusions per year. Her last infusion of packed red cells took place 30 days after CTX001 infusion. So at baseline, before infusion, patient 1 had a 3% fetal hemoglobin expression relative to total hemoglobin. Total hemoglobin was about 9 grams per deciliter, 3% of which was fetal hemoglobin. Now, after 18 months, that 3% expression of fetal hemoglobin turned into 93% expression of fetal hemoglobin. And pretty much from this point on, the patient never needed any more infusions. And again, this was a patient that was getting 16.5 infusions per year. And since this is a genetic disorder, these infusions had been going on from when she was born. So this is remarkable. Here's patient 2. Now, this isn't just some random patient. This is actually who patient 2 is. Her name is Victoria Gray. NPR, so many other companies have done interviews with her, written articles about her. She was a lot more open with the public about being the first patient treated with CRISPR for sickle cell disease. So yeah, Victoria Gray, if you Google her, you'll be able to read some really cool interviews, articles about her experience through the whole process. She was 33 when she consented to the trial. She had the homozygote phenotype, 2 sickle cell alleles. Before the study, she would experience 7 vaso-occlusive incidences per year, which led to 3.5 hospitalizations per year. And she would receive 5 units of packed red blood cells via transfusion per year. Here is the adverse event history, severe adverse event history during the trial for the patient. She experienced 114 adverse events, 3 severe adverse events in the 16.6 months of follow-up done for this table. Here are the grades for the adverse events. They chose to, the people who wrote the paper chose to list the most common adverse events for patient 2, whereas they didn't mention what the adverse events were for patient 1. So as you can see here, neck pain was very common, headache was very common, cholelithiasis, musculoskeletal and non-cardiac chest pain, esophagitis, stomatitis, lymphopenia, but it all resolved with treatment. The 3 serious adverse events the patient experienced were sepsis in the presence of neutropenia, which occurred on day 16, cholelithiasis on day 49, and then abdominal pain on day 56. They didn't give any more details for what the cause of the abdominal pain, what kind of abdominal pain it was, but the paper just mentioned abdominal pain on day 56 as a serious adverse event. So here are her results. Okay, so her last transfusion of red cells occurred 19 days after infusion of CTX001, and she had no vaso-occlusive episodes since infusion. At baseline, before infusion, she had a fetal hemoglobin percentage of 9% and sickle hemoglobin percentage of 74%. Then at Then at 15 months, 43% fetal hemoglobin and 52% sickle hemoglobin. And this composition was enough to lead to these results. No more vaso-occlusive episodes, no need for infusion. And what's interesting is what I found interesting was that 43% was enough. I mean, even here you're thinking, what is this? Like, if this is 9%, it's probably like 18, 20%. Anything above 20% seemed to be enough to just completely remove the need for infusion and the incidence of vaso-occlusive episodes. Something that I found kind of strange that they didn't talk about in the paper was this huge spike in hemoglobin A after infusion, and then just it dropping. Because over here you see that hemoglobin A, I don't know, 10%. Here you see it's nearly 90%. I don't know what I would estimate this as, but then it just diminishes, and they made no mention of it. So I don't know what to say about that, but it was something I caught eye of and thought was kind of strange. Here's a numeric analysis of the ratio of fetal hemoglobin to sickle hemoglobin. As you can see, fetal hemoglobin gradually increased over this 15-month period. Okay, so in conclusion, the researchers, they were able to show that CTX0001 had high levels of allelic editing and durable engraftment. High levels of that it was able to express high levels of fetal hemoglobin, and that it eliminated the incidence of vaso-occlusive episodes and the need for transfusion. One issue that both patients ran into with CTX0001 was lymphopenia associated with CD34 cell engraftment. And I did a little look into that, and CD34 cell engraftments are known to trigger a delay in immune system restoration. Other approaches towards bone marrow transplant with other cells often have quicker immune cell restoration, but at the expense of higher rates of graft for host disease, whereas CD34 has a low incidence of graft versus host disease. So I'm sure that's why they chose to use CD34. And some recent updates. So since this all started over 2 years ago, they've been able to administer CTX0001 to 8 more patients, and they have 3 months of follow-up data. And that data is broadly consistent with what they found with these 2 patients. So at this point, the primary future goal for CTX0001 is just to determine if it's generalizable to the public at large. All right, so that's my presentation. So now we can open it up to just discussions and questions.
I see some hands up, so I'm not sure who's went up first. I'm just going to start at the top of my list. Zach Zilla, please go ahead.
Thank you. I had a question about some of the slides where you showed that after treatment with CTX001, adverse effects increased in both patients 1 and 2, but the transfusions ceased. Because I'm just unfamiliar with this topic, why would transfusions cease if adverse effects increased after the drug was administered?
So my thought on that is, I'll move back to one of those slides. Yeah, we'll use this one. Is well, for one, in reporting of adverse events, anything that the patient reports is considered an adverse event. So if the patient reports neck pain, they can't prove that it's not related to the treatment, so it's going to be included as an adverse event. And also, the patients do have to undergo a bone marrow transplant where their entire immune system is knocked out, and it takes time for engraftment to actually take place. So during that period with the suppressed immune system, they're a lot more prone to various adverse events.
Okay, that makes a lot of sense. The immune system suppressed. Thank you for answering my question.
Thank you, Zach. I'll add to those adverse events. I don't think that would have prompted a transfusion, if that makes sense too, kind of, because I don't know, your question made me think that you were asking why wouldn't they get another blood transfusion if they were having adverse events, and I don't know if you were asking that or not, but I think they were kind of two separate, and Phil did a good job of explaining why the adverse events went with that drug specifically. Did that answer everything for you, Zach?
Yeah, I just didn't know, as well as the transfusions, if there's a protocol for stopping transfusions with packed red blood cells or not as well.
Okay, okay. And so let's see, Dr. Johnston, I see your hand up. Go ahead, please.
Yes, Phil, excellent review of that article. I learned a few things there. And I've got some questions, more questions than answers. So, and I'm just wondering if you ran across issues pertinent to this type of case. And I'll form a first question around iron. And I'll say that based upon the volume and the frequency of transfusions this patient had been subjected to, necessary for treatment. Had there been any reference in the labs regarding this patient's amount of iron that she had? Any notation?
So in the supplementary appendix for the paper, they included lab values, but they didn't include any analysis of lab values throughout the entire supplementary portion of the paper, including the actual manuscript itself. So when I looked at the lab values, I could only make my own assumptions, but because I didn't have any of their information, I didn't want to put anything into this presentation. I don't remember offhand if they monitored iron levels or included anything about prior iron levels before performing infusion.
That's a fair enough answer. As you well know, associated with transfusions, particularly multiple, can be hemosiderin depositions and iron load. And so I was just wondering if there had been any mention of iron involved here. The other question that I found, and I just saw one picture of this young lady, and the reason why I mentioned that is because you did discuss adverse effects. And under those was the timeliness of cholelithiasis. Now, did she undergo a cholecystectomy?
They didn't mention whether
she
went.
Go ahead, I'm sorry.
I was just going to say they didn't mention how they treated every adverse event, but with every adverse event, they stated that everything resolved with treatment.
Okay. The reason why I'm asking that, from that one picture, she looked maybe a little overweight, and maybe. So the likelihood of having a gallbladder attack in a setting of sickle cell, one of the culprits that can precipitate cholecystitis from stones is hemolysis. And you can have hemolytic issues that can cause abdominal pain. So you implied that everything spontaneously resolved. So I think as clinicians, you want to try to track some of those, hey, why did the lady have gallbladder symptoms, abdominal pain? And again, one of the components in a hemolytic scenario could contribute to acute gallstone attacks. So was there any reference to hemolysis?
That there really wasn't. I talked to
Okay.
Shelley about this yesterday. Like I was showing her lab values that they included in the supplementary appendix. There was a patient one actually had ALP at baseline around 80, and after 12 months around 160, and they made no mention of it anywhere in the paper. So there was definitely a big critique would be where they just left out. They left out a lot of information on the adverse events, the treatment of the adverse events, and the lab values that they included in the supplementary appendix, but then made no mention of why things went as they did throughout the 21 months of follow-up.
Okay. And the other question, the last, and I'll be quiet, is that you had implied that at one time she developed pneumonia, or one of the patients developed pneumonia in, I think, a post-treatment setting. Is that right?
Yeah, developed pneumonia following ammonia associated with neutropenia from the myoablative conditioning.
Right. My question, did they cite what kind of pneumonia? Bacterial, viral? They just
They did not.
okay. Okay. You would wonder under all these scenarios whether or not she would have been a candidate or would be a candidate, even at this early age, of pneumovax as a prophylactic treatment for further pneumonias to come. Okay, well, bottom line is all of these are spurred my interest to ask these questions because this is a very challenging case to manage. And if you didn't have exposure to CRISPR, you wouldn't really have much experience of how to manage these. We used to manage these with bed rest, fluids, and oxygen, and something for pain. And that was about it. And then we'd hope that the spleen or any other organ didn't infarct. And you just watch serial peripheral smears, reticulocyte counts, and hope that the symptoms would resolve. And a lot did. So I enjoyed your presentation. I thought you were very comfortable with your slides. You had great explanations of the content of your slides, and it was done very nicely. So thank you much.
Thank you, Dr. Johnston.
Thank you.
All right. Let's see. Amber McCormick, go ahead, please.
All right. After all of that, I have to remember what my question was. Lots of thought-provoking stuff going on there. I think it was about the the allelic editing frequency. It's been a long time since I've thought about CRISPR, but that's basically like how well it takes, like how well the CRISPR changes like have a lasting impact on the patient.
Yeah, so they would just look at that actual target sequence, the erythroid promoter sequence of BCL11A, what it's meant to look like, its wild type, or the original transcript before editing, and then what the transcript looks like after. And if they're not matching up, that's increasing the allelic editing frequency. So that's just what they're comparing. And in this case, they just wanted to place disruptions in there. They weren't trying to make anything accurate or targeted. They weren't trying to just make sure to hit just a specific cytosine or a specific adenine. They just wanted disruptions anywhere in that sequence. And then they would measure to see how much disruption there was comparatively to before treated.
Okay. Okay. Thank you.
Thank you, Amber. Okay, Dr. Wolf, go ahead, please.
Yeah, thanks. With hemoglobin S, I had always figured that the obvious target was the point mutation itself. So I was rather curious why you wouldn't go at the point mutation itself. That's my question number one. And question number two was, is it possible that they could have, rather than wiping out the entire immune system, just targeted the CD34 positive cells and repopulated them? Now, I guess you'd worry about potential autoimmune disease, but it seemed to me that some of the side effects that they were having here was because they knocked out the white cells and you are trying to alter red cells.
So to your first question, if could you repeat your first question first?
Yeah, sorry, I kind of get lost in my rambles here. The first question had to do if it's a point mutation and you got CRISPR, why would you not fix that point mutation in hemoglobin, the beta chain, rather than trying to upregulate the fetal hemoglobin?
I think their thinking there was that they had in previous research discovered that just a disruption in BCL2A will trigger an increase in fetal hemoglobin, and it doesn't have to be accurate, meaning they can use wild type CRISPR-Cas9, and it would be inherently safer since they didn't have to be as targeted in their approach. They didn't have to make sure it was at the exact base pair that they wanted to make the change, that as long as they could make a knockout somewhere in that promoter region, they would be able to raise the fetal hemoglobin. And so I think that's why they took this approach.
Okay.
And then could you repeat your second question?
My second question had to do with the fact that they had diseases as a consequence of neutropenia. And they did ablation, immune system ablation with their therapy there. And my question is, did you in fact have to knock out the entire bone marrow system, or could you just get by with knocking out your CD34 positive cells and leaving your certain other things unaffected? Would that have benefit? I'm not sure I worked through the logic of that, but that was what I was thinking, because what you're trying to replace is the white cell progenitor cells. I mean, the red cell progenitor cells. And to the extent that you've got white cells there, why target those that are in the process of dividing and maintaining your immune system during that process? That makes sense.
Yeah, can I, Phil, before you answer that, can I maybe clarify if you would know, Dr. Wolf, is there a way to target just that cell type, or when you knock down the immune system, is that a broad drug that's going to hit everything? Because they were given that drug beforehand, so that your question made me have a question, is could they have given them a different drug, I think is what you're saying, to target that different cell type versus wiping everything out before they got the transplant?
You know, and the answer to that is, I don't know. I mean, I think sometimes they just irradiate the hell out of everything and destroy it that way. With this drug here, I'm assuming it's a cross-linking drug, and so it's going to be active on cells that are dividing as a means of doing things. But that had me thinking, well, what if you you know, coated a CD34 positive cell with antibodies and kicked in your natural killer cells to come in there and kill them instead? Would you in fact circumvent some of the problems that were happening here?
Okay, so I'll start off with the true honest answer of I don't know, but what I think, what I would be curious of is what Shelley said, and it's that, is it possible to replace, because with a bone marrow transplant, you're effectively wiping out an entire organ system, as in you're wiping out the blood as an organ from the patient and giving them a new one. And by doing that, you're making sure that it's going to be uniform across the entire body as a whole. But when you're targeting one specific cell type, I kind of imagine you'd get some sort of chimeric phenotype where some CD34 cells will end up having the affected alterations where some won't. So you won't get an entire, you won't get a global effect as you would with the bone marrow transplant. But again, I don't know. I'm imagining the bone marrow transplant is just to obtain very global, stable results, as opposed to a more targeted approach that may not have led to as, because you're not entirely replacing the entire blood system in the body. Yeah, but I don't know.
Yeah, the comment in the chat, I was thinking along the same lines as if you could have like a rituximab, which targets the CD20. I mean, and you know, for that I always say it's like a B cell do-over, because you kill all your early stage B cells, but your plasma cells and everything are still on board, so you don't have the serious adverse effects because there isn't a period of nothing. And, you know, to the extent that you could prevent that period of nothingness, it seemed to me it was kind of an interesting thing.
I think it's another thought-provoking concept that if you knock them down and give them something, could you also give them a boost of something else so that they have those white blood cells to start, so they don't get the pneumonia, and maybe they don't get some of those other things that you were talking about, Dr. Wolf. So I don't know. I think it's going to be evolving. I mean, with this technology, my conversation with Phil was really, I mean, look at the demographics and the populations of people that suffer from these disorders. Phil did a good job of pointing out that they suffer from their whole life. I mean, from 3 months old, they're having side effects. They're receiving blood transfusions, 16 every year. Could you imagine the cost, the debt, the impact on your life, whether you can work, whether you can afford things in the future because your healthcare bills are so massive? And so a therapy like this that you could give one time, one and done, and I don't have to go back and get those 16 transfusions times 70 years because I'm going to live to be 89. I imagine there's a big cost benefit there, but where medicine and technology has to get is it has to be affordable. So at what point are we going to get this to where the people that need it are the peoples that are in the most kind of disparaged situations and have least access to healthcare, and they're the ones that need the help. So I think it's a huge thing that's going to be in medicine forever, and CRISPR, I hope, makes these things more readily available for these people. Dr. Johnson, go ahead.
Just a comment. No wonder she was, and I think, Phil, you had referenced positive for Hep B with the host of multiple transfusions. Did you say she was B positive?
She, patient 1 had a history of viral hepatitis C.
Oh, I thought you said B. I'm sorry, I misunderstood. Okay.
No, that's fine. I wanted to add to what Shelley was saying. I looked up some numbers before this presentation on the actual cost of a bone marrow transplant or a one-time CAR T-cell therapy. So for a CAR T-cell therapy, and reason I'm saying CAR T-cell bone marrow transplant, because I feel like this CTX0001 treatment is very similar in what's going to be happening to the patient. And the cost of one treatment of CAR T-cells is about $375,000. A bone marrow transplant will cost between $100,000 and $300,000. And then the annual average cost of treating a patient with sickle cell disease is $30,000. So in just 10 years, you're already equaling the cost of treatment at the high end for a bone marrow transplant, and you're nearing the cost of treatment for a CAR T-cell therapy. But you're only going to do this once on the patient, and then from there on, they don't need to be treated. So in terms of generalizability, it does seem like something insurances would begin to consider, because in the grand scheme of things, they're going to end up saving a lot of money on patients with this disease within 10 years. And if it starts at birth, yeah, it'll be pretty quick. It's pretty easy to see economically why this could be something very generalizable.
Thanks for getting that info, Phil. I appreciate that. I know, Zach, you've got your hand up again. Go ahead, please.
Thanks. Your discussion earlier of being able to have that specificity of wiping out a specific type of blood cell, either red or white, in order to decrease the amount of serious adverse events or adverse events with regards to that immunological response. If you had that specificity with a drug, wouldn't that also help with a lot of other debilitating diseases with the bone marrow and blood, like leukemia? So like, we haven't come close to even having that specificity with any sort of drug on affecting specific blood cells.
Okay, so I'll try to answer this as best as I can. In terms of specificity, that's always priority number one, because they primarily off-target edits could be deleterious and lead to a lot of issues down the road that we don't even know about. So with CRISPR-Cas9, at least in other models, non-human models, edits are made with very, very high accuracy. But I think because this was the first real study using CRISPR in humans, their goal was to do this in the most ethical and safest way possible. And that's why they did it in this approach, where they're doing something without affecting germ lines in any way. And they're using the only organ system in the body that can effectively be removed and put back in, and making the changes uniformly. Whereas like if you're looking at maybe like Duchenne's muscular dystrophy, for example, if you were to make edits to muscle cells, you would have to make the edits to every muscle cell throughout the entire body. Otherwise, you'll end up with like a chimeric state where some cells are resolved of the muscular dystrophy and other cells are still dystrophic. So I think that's, I feel like I actually kind of turned away from your question, but hopefully that was a decent answer.
No, it was. I don't think, I mean, if we had something that was so specific, I think that would, in addition to solving TBDT as well as sickle cell disease, I think it had the generalizability with regards to pathology would be huge if we could specific attack.
Well, I will say that this is a breakthrough for one reason, because it's shown that CRISPR can work in humans in an ethical manner, that we're not doing any embryonic edits or germline edits. 10, 20, 30, 40 years down the road, maybe we begin doing that, but it's opened the door to using CRISPR on humans. So more specific edits could be done later down the line. But for now, they did it this way and it worked.
Regarding your comment about leukemia, I mean, the challenge there is that leukemic cells have escaped our immune system. They don't have antigens on their surface that are detected and are recognized as foreign, and therefore our immune system doesn't destroy them. And so that creates a challenge there, and then they're heterogeneous as well. So it's not like they all express the same alternative proteins there, but certainly when cancers do express foreign proteins, and they do so with any reproducibility, that creates then a target. So the HER gene for breast cancer, for example, are those things where there are things that are specifically targeted because they show up with frequency, but it's that lack of frequency that makes cancer a bugger to treat.
Well, thank you, Dr. Wolf, and thank you, student doctors, for coming, and everybody for participating in the discussion. I think it's been really great, and thank you, Phil, for bringing it here. I'm going to say, please look for your announcements, come back when you can, and have a nice weekend, everybody. Thank you for coming. We appreciate you all.
Thank you guys for coming. I appreciate it.